Hey all. Okay, so this was a little bit rushed in SI, and I just want to make sure everyone gets it. So, basically, what we did was we said there's four little kind of categories that we'll have on screen here. And I'm gonna actually move all of these over because I messed up. There we go. And in these four categories, we're gonna write the four steps of cellular respiration, right? So we have glycolysis, we have pyruvate oxidation, and a lot of you might actually also know this as the pyruvate dehydrogenase complex, or PDC. It's the same exact thing. We usually just call it pyruvate oxidation, though. Here you have the Krebs cycle, or the citric acid cycle, or the TCA cycle. It's all the same thing, right? And at the end, you have the electron transport chain. So the point of this exercise is to think about it in numbers, right? Think about it in numbers and think about it in ability. So there's a few things to ask here. First of all, first thing I'm going to ask is, where does this happen? And it's really about making sure you guys understand what can do these things. So everything, every living organism needs to do glycolysis. Everything that's alive. There's prokaryotes, eukaryotes, everything in between, everything does glycolysis, right? And glycolysis is literally glycolysis, right? It is glucose splitting. Glucose split. You're cutting a glucose in half and you're getting energy out of it. Every living thing does this. And here's what I had you guys do in SI session. First of all, I had you guys identify where this happened. And because every living thing does it, it has to happen in the cytosol. Cytosol, cytoplasm, same thing, right? Whatever you guys call it. It has to happen in the cytoplasm because every living thing does it, and not every living thing has the other structures that we need. Every living thing has a cytoplasm, though. So we need to do that in the cytoplasm. So what goes into glycolysis? I actually just said it. Clues in the name. A glucose, right? So one glucose, one, sorry, not yet. This one glucose is the fuel for glycolysis, right? It's in the name. It's where all the energy comes from in the first place, right? It's the fuel. At the same time, glycolysis also needs an input of 2 NAD plus, right? Another input is 2 ATP. Okay, so now what? Well, glycolysis, its purpose is to split sugar, right? So this glucose, which is actually a 6-carbon molecule, is going to get processed and split into 2 pyruvate. Pyruvate, a 3-carbon molecule. And you can do the math there. 1 times 6, this is the same as 2 times 3, right? No carbons are lost. NAD plus will go 2 for 2, turning into NADH. The proper way to write this is NADHH plus, but most of you will just see NADH. ATP gets processed and actually ends up spitting out 4 ATP. This is a big process. It's kind of interesting what actually happens, but just by the numbers alone, 2 ATP turns into 4 ATP. Now, a big learning goal of this is to understand these things as big redox reactions, okay? Glucose to pyruvate is an oxidation. In return, NAD plus to NADH is a reduction. These two things happen at the same time. All redox reactions come chained, right? One oxidation has to happen with one reduction. And these are the oxidation and reduction of glycolysis. So there are your numbers so far. It's going to be 1 glucose, 2 pyruvate, 2 NAD plus, 2 NADH, and then 2 ATP in, 4 ATP out. If you're a bacteria, if you're a prokaryote, you cannot continue onto these other things because you don't have a mitochondria. These things are aerobic processes. They happen in the mitochondria. Glycolysis is an anaerobic process. It happens in the cytosol, right? And so to continue to do all these things, you need to have a mitochondria. A mitochondria itself, mitochondrion, is a prokaryote. So only eukaryotes really have them. If you're a prokaryote, if you're a bacteria-sized, you're physically smaller than a mitochondrion, so you don't have them. And you can't do all these processes. So then you're kind of at a dead end, right? You're making pyruvate, you're making NADH, and you're making ATP. Great. But what happens if you make all of your NAD plus into NADH? Right? What happens when you run out of NAD plus and you can't continue this process? Well, there's a special little process here that bacteria will undergo in anaerobic conditions called fermentation. And actually, this is ethanol fermentation. So in fermentation, we're going to take the output from glycolysis, and I want you to see this as something that happens all the time. The output is going to become the input, right? So pyruvate to pyruvate. What else did we output? NADH to NADH, right? And these are going to become 2-ethanol and 2-NAD plus at a cost of 2 ATP, turning it to ADP. Now let's run the math really quickly. We've created 2-ethanol from 2-pyruvate. So this is where we got our 2-pyruvate at the end of glycolysis. We go into fermentation with our 2-pyruvate. We go into fermentation with our 2-NADH, and we create 2-NAD+, which is ready to be used in glycolysis right there. There's that connection right there. So this kind of turns into a cycle, which is exactly what bacteria do. They do this cycle. The problem is, if you do the math and you cancel the fact that you put into ATP, right, and you say that the net of glycolysis is 2 ATP, right, because you put into, got 4 out, took 2 home. If you say the net of glycolysis is 2 ATP, well, you just use your 2 ATP. So you're done. Your net is 0, right? So bacteria will go in this endless cycle of glycolysis and fermentation, and they suck, right? They can't get big. They can't develop past single-celled. They kind of suck, which is why bacteria are so small. However, you're in luck. You have a mitochondria, and you can continue on this process, right? So let's do what bacteria did, but better. We're going to start with 2-pyruvate. Okay, we're going to start with 2-pyruvate, and we're going to turn this pyruvate into a molecule called acetyl-CoA. Now, acetyl-CoA is a 2-carbon molecule, but pyruvate is a 3-carbon molecule. We've gone from 6 total carbons to 4 total carbons. We've lost 2 carbons on the way. The way this happens is CO2 leaves the body. And this is part of where you exhale. You exhale CO2. This is one of the places that happens, right? Pyruvate oxidation also requires 2 NAD+, one for each pyruvate, turning into 2 NADH. Now remember, what we said is that everything is a redox, right? Glucose becomes oxidized in pyruvate, NAD+ becomes reduced, right? Fermentation, same thing happens. Pyruvate, through a series of steps, becomes reduced into ethanol. NADH becomes oxidized into NAD+, right? So this is actually an oxidation, and this is a reduction. Notice that whenever we're generating NAD+, it's an oxidation. But whenever we're generating NADH, it's a reduction, right? And actually, let me just correct this really quickly. There we go. So therefore, this must also be a reduction. It's the same reaction as we've seen before, right? Two are the same reaction. And you'll actually see that this happens a lot. But in return, this must be an oxidation because it's one of two reactions that happen. If this is a reduction, this must be an oxidation. And look at that, pyruvate oxidation. There it is. Pyruvate is being oxidized into acetyl-CoA. Right? So these things are named these things for a reason. Moving on, we're going to take this acetyl-CoA that we just made, and you guessed it, it's going to be the next input. Acetyl-CoA will go into the Krebs cycle, turning eventually into 4-carbon dioxide. Now, this is technically an oxidative process. Let's do the counting here, right? 2 acetyl-CoAs, 2 2-carbon molecules are turning into 4 1-carbon molecules, right? And so this turning into this, oxidation. In return, we bring back our old friend, except a lot bigger in scale. 6 NAD plus going to 6 NADH, right? And then we also include one newcomer, 2 FAD, going into 2 FADH2. Now, you might be asking, what is the difference between NADH and FADH2? That is biochemistry. That is organic chemistry. For you, there is no difference. They are both high-energy electron carriers that actually serve as the reductions of this oxidative reaction. There's your redox. Now, every single time here, an input has been an output. An input has been an output, and we've kind of been recycling all of these things, except this, and this, and this, and this. We've generated these high-energy electron carriers in every single reaction thus far. Yes, we've also generated ATP, but that's just gone away into the cell. In fact, the citric acid cycle also generates 2 ATP. But those just go away in the cell, right? ATP is used everywhere. It's cache, it disappears. We haven't used these anywhere thus far. Where do you think we're going to use them next? The electron transport chain. Into the electron transport chain goes 6 NADH+ from the Krebs cycle, 2 NADH+ from pyruvate oxidation, and 2 NADH+ from glycolysis for a total of 10 NADH+, 2 FADH2. Let me actually give that a little bit more space. And oxygen, because you need oxygen to breathe. So we're actually going to say 1 oxygen, and we'll see where this is going. Finally, we are able to turn all of these electron carriers into their oxidized form, right? 2 FAD, 10 NAD+. Oxygen will actually accept those electrons and become water in your cell, right? These numbers for oxygen don't really matter, but these numbers do matter, and let me show you something. 10 NAD+, 2 FAD, where are they going to go back? Right here, and right here, and right here, and right here. It's a cycle. It's a very big cycle, but it's a cycle. Bacteria has a much smaller cycle, but it's still a cycle, right? They still produce NAD+. You just took a lot longer to do it. Finally, electron transport chain generates 34 ATP per glucose. How? Biochemistry, but it is 34. You gained 2 from the citric acid cycle, you gained 2 from glycolysis, you gained 34 from the electron transport chain. Net per glucose, 38 ATP. This is cellular respiration by the numbers.